Good evening, everyone. So, I just want to remind everyone quickly that among the two classes which were going to be for free, today's class is the second one. So, this is the final class when you can attend it in live, in person for free. And after this, all the classes will be only available for the people who have registered for this course. And if you want to register, that is still possible. All the information and details are available in the description. Also, since we have a few minutes before we begin, I would like to remind everyone who has joined today, the registered participants, that I have uploaded assignment, the first assignment already via the Google Classroom. I hope everyone has had time to have a look at the Google Classroom and have joined it. The assignment can be completed on or before 30th of this month, 30th of July. And if you complete all the assignments, only then will it be possible for you to receive the course certificate. Also, from today's session, there will be two assignments. They are not going to be super complex, but I would request everyone to go through it, go through the session if needed, and complete the assignments on time. Since they are very easy and do not require a lot of time, it would be much easier for you to complete them day by day rather than keeping it for the last day. So that's my personal suggestion. Please have a look at the assignment section after the lectures. So to all of the people who are watching live, once again, this is going to be the final day where you can watch this course for free, attend this course for free. After today, only the registered participants will be able to attend this course. In case if you are interested to join this course and continue learning more about molecular biology, which is essential, the description for registration is there in the information below the link where you have been attending this live session. So someone from the registered participants, so Darshana has asked the question. So no, the as I clarified yesterday, whenever a DNA sequence has been mentioned, it is always mentioned, and it is correct to assume that it is in 5 prime to 3 prime direction. So you cannot assume that the direction is from 3 prime to 5 prime. So yeah, you have to consider that it is from 5 prime to 3 prime. Good evening, Akshat. Good evening, Sudarshana. All right, guys. So I think all the registered participants have joined today's session. So today's session is very, very critical for you to understand the molecular biology. And so I would say that you all should pay utmost attention to today's session. It is very much in detail. I have tried to simplify each and every concept that I will be teaching today. And I would request everyone to go through the PowerPoint presentation as well, which I will be providing on the Google Classroom. so that you can revise this session before you attend the next session. All right, so let's begin. So today we are going to talk about the molecular translation of life. So more about the RNA and the protein rather than the DNA. We have already discussed about a lot of details of the DNA yesterday. Today we are going to discuss a bit about the translation part and the transcription part. So let's begin. To give you an overview, we will first revise the central dogma of life briefly. We will see how does DNA get translated to protein, then the critical components which are essential for translation, what are mutations and what are the types, some hands-on exercise, and then I will summarize the session for today. All right. So I would like to quickly go through and revise yesterday's concept which we learned, the central dogma of life. We saw that the flow of the process is from DNA to mRNA to protein. And if you have a look on the right hand side, this is also the picture which I showed you yesterday. So first the DNA is transcribed to RNA, and then the RNA is translated to the protein. And there are different roles of different specific enzymes which are involved, and in case of translation the ribosome is involved. Of course there are also some enzymes here, no doubt about it, but we will have a look at it more in detail as we go ahead. So we also know that DNA is a complex biochemical moiety which carries the important information in order to build the molecular basis of all the organisms we see around. We explored transcription, that is the process of getting to RNA from the DNA. So today's focus will be on the translation part, that is how does RNA get to the point of having amino acids and form the protein, and how does the cellular machinery read the code. All right, so first let's have a look at the translation right from the beginning. So what are the parts of the translation? So once again, for your reference, I'm stating the central dogma on the left hand side. For comparison, you know, biology is more complex than we can imagine. This is just a very simplified image of whatever takes place inside a cell. So right now I am trying to show you a little bit more detailed, I wouldn't call it complicated, but more detailed part of this process which has been simplified here on the right hand side. So do not fear after having a look at this particular diagram. Just have a look at it step by step and try to focus on the points I am trying to tell you. So let's have a look at the DNA first. So DNA has different regions, although this has been shown you with respective nomenclatures like promoter, exon, intron, template strand, etc. When you see a DNA, you are just going to see a DNA. There is no labeling as such inside the cell. This is just for your reference and simplification. Then comes the transcription part, wherein you get the pre-mRNA first, and then after the RNA processing, you get the mature mRNA. So right now, what we would want to focus on is different regions of the DNA and mature mRNA and what are the different functions. So this was just a summary picture. I will go through each and every component which is essential for us to learn in the molecular biology. All right, so let us go through some concepts. So right now I would like you to focus on the promoter region. So this particular part which has been highlighted in green, what is it? So promoter is a regulatory DNA sequence which is located on the left hand side or the upstream region of the gene's transcription site. So this transcription unit is typically termed as gene. So gene is going to be the part wherein there will be exon and intron. So this is the gene, and upstream to it, meaning on the left hand side of it, is the promoter location. So what is the function? The function is that it serves as the primary binding site for RNA polymerase. I hope you remember that the RNA polymerase is the enzyme which is required to convert the DNA to mRNA. and it is also important for some general transcription factors to bind. Since these transcription factors are not as relevant as the other parts for us to get through the molecular biology, I'm not going to go through a lot of details about the transcription factors. So the promoter serves as on or off switch that decides when, where, and at what baseline level or minimum level a gene is transcribed into RNA. So the promoter, its presence is very, very important or critical in a DNA sequence. Now, let's have a look at the other parts. So right now, this is the last part of the diagram which I showed you before. What I would like you to focus on is the UTR. One I have highlighted inside the red box. So 5' untranslated region, or the UTR, is the part of the mature mRNA that begins at the transcription start sites and it just ends before the start codon. So as you can see, this is the transcription start site, okay, and this is the start codon. So it is just ending before the beginning of the start codon. It is transcribed into RNA, but it is not translated into protein, meaning that actually the UTR region will not contribute towards formation of any amino acids or proteins. What is the function then, you might be wondering? So it is important for regulation of translation. It contains critical recognition sites for ribosome binding, because you know that ribosome is important for translation. It has Kozak sequences in eukaryotes. So we will learn what is a Kozak sequence ahead. You do not need to by heart the Kozak sequence. You just need to know that its presence is very critical. So in case of eukaryotes, this particular sequence, which is part of UTR, is called Kozak sequence. However, in case of prokaryotes, it is known as Shine-Dalgarno sequence. So just for your knowledge, and it frequently forms secondary structures like hairpins. So these are complicated structures of RNA which have different functions, and this UTR influences mRNA stability and how efficiently the ribosome can initiate translation. All right, moving ahead, let's talk about start codon. So this particular green part which has been shown here is the start codon. Typically it is AUG. It can be a bit different in case of prokaryotes, but also normally what has been observed is it is AUG. So it is the first triplet of nucleotides. So triplet meaning they are always going to be present in set of 3 nucleotides, and these set of 3 nucleotides is called as triplet, which is also known as codon. So the first triplet of nucleotides in the mRNA transcript that is translated by a ribosome is the start codon. It is very important because it is kind of saying that if the start codon is not present, then the whole process might be abandoned. Okay, so the function is to signal the exact location for the ribosome to begin synthesizing the polypeptide chain. Polypeptide is nothing but protein, simultaneously establishing the correct reading frame. So reading frame meaning that this ribosome will need to know which is the part of the DNA through which I have to produce amino acids, okay, or add amino acids. So it determines the correct reading frame for the rest of the sequence. It almost universally codes for the amino acids methionine. So this is a very important and crucial fact. So almost all the times your DNA sequence which has AUG in the beginning, when it translates to a protein, you might almost always find that the first amino acid is going to be methionine. All right, so let's have a look at the coding sequence, or the CDS. This is the most interesting part for all of us, because when we talk about a gene, we forget about all these components which are around it. We are just talking about the CDS, or the coding sequence. But as you will now know ahead, that other regions are also very crucial. But for now, let's talk about the coding sequence, or CDS. Coding sequence is the central portion of mRNA which is derived from spliced exons of the DNA that contains the actual genetic blueprint of the protein. So what do I mean by splicing? you saw that the gene actually contains introns and exons, introns and exons, introns and exons as alternate parts. So exon is the part of the gene which actually encodes for the amino acids, okay? And intron is something which is removed. It is not going to be translated to a protein. So of course, you have to remove the introns which are present in between the exons. And when this is done and the exon regions are joined together, this process is known as splicing, okay? So this is why it is written here that it is derived from spliced exons. So when the exons are joined together, you actually get the CDS or the coding sequence. And what is the function? It consists of a continuous sequence of codons that dictate specific linear order of amino acids. Very logical, right? Because when you have all the exons together, it will naturally code for all the proteins because the introns have been removed. And these amino acids then come together during the translation to form a functional protein. All right. So now let's talk about stop codon. So stop codon, as the term dictates or says, is a specific nucleotide triplet at the end of the coding sequence that does not code for any amino acid. So why is it present? It is present to tell the ribosome that okay, now the job is done. Now you have to stop the process wherein you are considering that the triplet which is present should code for an amino acid. So here it is like telling the ribosome that you should stop here. So it serves as the termination signal for translation. When the ribosome reaches a stop codon, the release factors bind to it. So release factors are typically different enzymes and set of cofactors which are going to participate. We are not concerned with them right now, but yeah, they also bind to the stop codon, and this promotes the ribosome to actually detach from the mRNA and release the newly completed polypeptide chain. So if the stop codon is not present, the termination won't happen and it will be a continuous process wherein the amino acids will be produced, and we do not want this to happen. So the stop codon is a crucial part. Now let's discuss about the 3' untranslated region. So this is the part of mRNA that immediately follows the stop codon and extends to the poly A tail. Like the 5' UTR, it is transcribed but not translated. All right, just so you know, I told you that the UTR is present on the upstream of the start codon, which means start codon is here, and on the left hand side, anything which is present on its left hand side, it is called upstream, right? So anything which is after the start codon will be called as downstream. So the coding sequence is downstream to the start codon. The stop codon is also downstream to the start codon. The UTR, 3' UTR, is also downstream to the start codon. So just don't get confused when I am talking about upstream and downstream. So I hope everyone is clear with the term upstream and downstream. Okay, it is always relative, it is not fixed. Whenever you are talking about upstream or downstream, whenever you use it, it will be always used in reference to some region. Okay, so what is the function of 3' UTR? It plays a vital role in the post-transcriptional gene expression. It contains regulatory sequences that determine mRNA stability. So, you know, the mRNA cannot remain inside the cell for unlimited time, because if it remains for unlimited time, the proteins will be continuously produced, which we do not want to happen. So the mRNA is supposed to have a limited lifespan. So the 3' UTR will determine how long the RNA lasts before degrading. It also decides whether mRNA is localized within the cell, which means the subcellular location of the whole mRNA will be determined by the 3' UTR, and how efficiently it is translated. It is also a primary binding site for regulatory molecules like microRNAs. So you can read more about miRNAs if you are interested. Right now, this is just an accessory information. We do not need to know more about microRNAs for the basics. All right. And now you might have observed there are two more sequences or two more components which are present in the mature mRNA. So all students who have been attentive so far can anyone point out which are these regions which we have still not talked about? Yes, Sudarshana, it is 5 cap and 3 prime poly A tail. You're absolutely right. So others also make sure that you participate so that you gain most out of these lectures. Okay, just try to be attentive. So the 5 prime cap is something we have not talked about, and poly A tail also is something we have not talked about, but they are very critical. So what is the function of the 5 prime cap? So 5 prime cap is modified guanine nucleotide which is added to the 5 prime end of mRNA. Its typical function is to provide protection to the mRNA transcript from the degradation by exonucleases, and it is also recognized by the ribosome as a binding signal to initiate translation. Um, this is all like very typical stuff from molecular biology, and you should revise the lecture which we are going through today. But I will tell you an interesting fact. Like you can go and read about 5 prime cap and its role in the influenza virus replication, just if anyone among you is interested. The influenza virus, when it enters the cell, it actually has something called as cap snatching mechanism. So it will actually use the cap of the host. It will cut it from the host RNA, mRNA, and it will use it on its own RNA, so that its own RNA cannot be recognized by the host immune mechanism and will not be degraded. So that's like so smart. So I just thought about mentioning this fact here. So influenza has this cap snatching mechanism. Now talking about the poly A tail, it is a long stretch of adenine nucleotides which is added to the 3 prime end of mRNA. It protects the mRNA from degradation, and it helps in the export of mature mRNA from the nucleus to the cytoplasm and plays a role in the translation initiation. So, the poly A tail will determine how efficiently the mRNA moves from nucleus to cytoplasm, because it cannot be present in just one location. So, as and when it's needed, it should be transported to the cytoplasm. So yeah, both these components are also very important. All right. So now let's have a look about the ribosome binding sites, or the RBS. So the ribosome binding sites are different in case of prokaryotes and in case of eukaryotes. So talking about the prokaryotes, how is it different? So prokaryotic mRNA binding uh, is involving all the different subunits, among which the small ribosomal subunit is one part, the Shine-Delgado sequence is another part, and the 30S subunit is also an important component. As you might have observed, the Shine-Delgado sequence is present in case of prokaryotes. So this particular sequence is very important and will determine the efficiency of the translation. Okay, so as you have observed, the small ribosomal subunit will come and bind to the Shine-Delgado sequence, and along with the fmet tRNA, it will actually go and bind and recognize the triplets which are present, triplet codons which are present inside the mRNA here. Okay, so in case of prokaryotic mRNA binding, it is a direct process, direct binding. So the small ribosomal subunit 30S binds directly to the mRNA strand. The target sequence, um, so it recognizes and binds to a specific region called Shine-Delgado sequence, and it is almost always important. So remember this. You should remember this point, because it will be talked about again when we learn about molecular cloning. So Shine-Delgado sequence is very important. There is no scanning which takes place. The ribosome positions itself exactly where the translation needs to start, and there is an initiator amino acid which is present. So the initiator amino acid is typically called formyl methionine. The starting tRNA carries a modified amino acid, that is fmet. Okay, so this is something you should remember. Two things: Shine-Dalgano sequence and about the fmet tRNA. So regarding the other parts, small ribosome subunit and the 30S subunit, of course these are important, but this is not something which you will encounter in molecular biology cloning ahead. All right, now let's talk about ribosomal binding sites in case of eukaryotes. So it looks quite different, right? So what you observe here is a number of things. First of all, it is not the fmet or formyl methionine which is present with tRNA, it is methionine, just methionine. Okay, and we saw earlier that there is no 5 prime cap which is present. Okay, but here in case of the eukaryotic mRNA, 5 prime cap is present and poly A tail is present. The subunit, the larger subunit is 40S subunit, and then after the scanning, this is the sequence of binding which takes place. So it is cap dependent. The small ribosomal subunit 40S cannot bind internally, it must attach to the 5 prime cap at the end of mRNA. There is always scanning mechanism. So once it is attached, the ribosome scans or slides to the mRNA until it finds the first start codon. So start codon is very important. Initiator amino acid, it is always going to be methionine. The starting tRNA carries the standard methionine. And what is the structural aid? Efficient binding and translation are heavily dependent on both 5 prime cap and the poly A tail. So as I mentioned, we saw that both of them are present in case of eukaryotic mRNA but are absent in case of prokaryotic mRNA. Now let us revise what are the key factors for ribosomal binding sites. They need a specific signal to attach to the mRNA and begin translation correctly. Shine-Dalgano sequence is the ribosomal binding site in prokaryotes, for example bacteria. They are located at the upstream of the start codon. In case of eukaryotes, the name is differing and the sequence differs a bit. It is called as Kozak sequence. It surrounds the start codon and helps the ribosome identify the correct initiation site. So always remember, without these, translation is inefficient or fails entirely. So what do you mean by inefficient? That you will not get sufficient quantity or amount of protein which will be produced, because um, there is no mechanism wherein you tell the ribosome that okay, I want this protein to be produced in such and such quantity. Okay, so this will be highly inefficient or slow, or it might also fail entirely. Now let's discuss about the codon concept. So you might have seen this table in biochemistry or biotechnology textbook. But what are the key concepts in this? Do not try to look on the table. I will try to explain the table shortly. Have a look at what I am trying to show you. So the genetic code is read in triplets, and the triplet is known as codon. So three bases, when they are read, it actually translates to one amino acid. So remember, when you are seeing three bases, it is actually only going to give you one amino acids upon translation. What is the start codon? AUG, which is coding for the amino acid methionine. This is the green light for translation. So without this, the translation will not begin. And stop codons are one of the three. So UAA, UAG, or UGA. So they do not code for an amino acid. They are just going to tell that, okay, now is the time to stop. And they act as red light, signaling the ribosome to detach and release the protein. All right. So now coming to this particular wheel, the codon wheel, how do you read it? So in order to read it, you go from inside to outside. Okay, so here they have mentioned 5 prime because of the direction. So this is from 5 prime to 3 prime. Always remember, like I taught you yesterday, the DNA sequence, RNA sequence, it will be always read in the 5 prime to 3 prime direction for convention purposes. So first you have to read the first amino acid here, which is U, and then you have to move outside. So if you want to know what is going to be the amino acid which is rising from the triplet codon U, and U. So if you have U, and U, it will be phenylalanine. Okay. If I have U, and C, it will be still phenylalanine. But if I have U, and A, it will give rise to leucine. If I have U, G, and G, if I see this U, G, and G, it will give rise to tryptophan. But if you have U, G, and A, this is red dot, meaning it is a stop codon. Okay. Now you can also have a look for the start codon here. If it is going to be A, U, G, okay, A, U, and G, what will it give rise to? Methionine, which is what? It is a start codon, as we saw earlier. So one of the interesting things for you to practice would also be, after you get this presentation, just go through it and try to read which amino acid is translated. So which amino acid is produced when you have a particular codon. Okay, just try to go through it. It is very interesting. So now let's have a look at what happens when the code changes. So essentially we need to learn about mutations. What are mutations? So mutation is nothing but a permanent change in the nucleotide sequence. So it is a permanent change, remember this. Now the mutations can happen at different levels of the base. So the first is substitution. So when a base is swapped, so one base is swapped for another, it is called substitution. So as the English term dictates, substitution means replacing something with the other. So swapping one base for another. Deletion means removing a base. So ideally, if there are four bases and one of them is removed, it will be termed as deletion. The mutation will be called as a deletion. Third type is insertion. So when you have a sequence wherein, for example, there are five bases which are present, A, T, G, and C, and an additional base is inserted when the mutation takes place. So for example, if one T is inserted additionally, if the sequence is A, T, T, and G, and C, then it is an insertion. Of course, you do not have to imagine all of this. I will be showing you a visual. Right now, just focus on the definitions. So insertions and deletions are commonly known as indels, and they can cause the entire frame shift. Okay, of course, and then this changes how the subsequent triplet is read. Now to make you understand this in easier terms, I have some example here. So this is the normal or the reference sequence. So this is the peptide, meaning the protein which is produced. So a short protein will be called as peptide. So these three will code for methionine, these three will code for threonine, these three will give rise to glycine, and then these three will be phenylalanine, and then UAA, which is a stop codon. This is wild type reading frame. But when then there is a substitution, that is missense. So for example, the original sequence here had G, but in place of G, when there is an A, the amino acid will change. So you can refer to the earlier codon wheel that was provided and just try to see. So when G changes to A, then of course the whole codon has been changed. So it will be not G, G, and T, it will be A, G, and T. So that will give rise to serine. Okay. So what does this translate to? So this causes missense mutations which alters one amino acid. Now if the amino acid is having similar chemical property, then it does not affect the protein so much. But if it has a different base property, so for example, it is an acidic protein which is being substituted by a basic protein, then of course the properties are going to be changed. Now second type is the insertion or the frame shift mutation. Okay, so what happens here is ideally the sequence should not have an additional base which should be present. So if this happens, what is going to happen? So for example, here the original sequence ATG, ACA. But here there is an insertion. So when an insertion takes place, there is an additional base which is inserted. So this will again change the amino acid. Okay, so the amino acid is changed, and again the problem is that because the frame is shifted, you do not have the availability of the stop codon. Did you realize that there is no stop codon present here now, because the entire frame has shifted, because the code is always read in triplets. So as a consequence, the frame shift mutation results in non-functional or truncated protein. All right, now let's have a look at the deletion. So when it is deletion, it also results in frame shift. And for example, here they have shown that the A is removed. Okay, so what happens then is that you get another sequence. So if there is a deletion, one of the base is lacking, and this will again frame like shift the frame of the bases. So this will again lead to different amino acids which are produced. So methionine, lysine, valine, and phenylalanine. So did you see how much difference it has made from the original wild type protein? So methionine, threonine, glycine, phenylalanine. Substitution will change this to methionine, threonine, serine, and phenylalanine, and so on and so forth. So there are a lot of changes, and then the stop codon is also not available. So this is bad. Like this is not what ideally should happen in a cell. So how does it affect on the protein level? How mutation make changes to the proteins? So there are again different types. So there are silent mutation. If the base changes but the amino acid remain the same due to codon redundancy, then it is not going to affect. As you saw earlier, that more than one codon can give rise to the same amino acid. Okay, so this is what they are telling here. If the base of the DNA changes but the amino acid remains the same, then it is going to be silent mutation. One might not even come to know about it unless they give the DNA for sequencing. Then there is second type, where it is missense or altered mutation. So the base change results in a different amino acid, potentially changing the protein folding and function. So as I told you before, if it is going to be a base which is resulting in a totally different amino acid with a different property, this can happen. And then there is another type which is called nonsense mutation, which is also known as something having premature stop. So this is where the mutation creates an early stop codon, resulting in a truncated, usually non-functional protein. All right, let's have a look at some examples. So this is the wild type or the reference gene. Okay, now this is the mRNA, and we have a wild type peptide, which is methionine tyrosine phenylalanine. When it is a silent mutation, when the T changed to U, it did not give rise to any other amino acid because it was codon redundancy, which means more than one triplet can give rise to the same amino acid. So it did not change anything. So it is completely like the wild type. Now the next is the missense mutation. So when uh, base is replaced, so for example in case of the wild type, it is going to be TAC, and in case of the missense mutation, it is CAC. So this will change the codon, and this particular codon gives rise to histidine. So then the amino acid is changed here. So one amino acid is substituted, and then it codes for the different amino acid. It has changed the amino acid change: methionine, histine, phenylalanine. And the nonsense mutation. So for example, it should have been, what should have been? It should have been the TAC, as you see in the wild type, but now instead of C, it is A. So how does this change? So in case of mRNA, it translates to UAA, and we all know that UAA leads to stop codon. So if the protein translation is stopped here, there is no question of the next amino acid getting inculcated or integrated. Okay, so this is premature stop codon because this has stopped before we want it to stop. So this is nonsense mutation. All right, so now something exciting where we can all interact a bit. um, Now I would like you all to apply a little bit of your knowledge you have gained so far. So I'm giving you two sequences, okay? So this is the first sequence and this is the second sequence. Now I would like you to identify the two gene sequences below and tell me if both of them are capable of coding for a functional protein. And if they are not, you have to tell me why they are not able to do so. Like what is the flaw in the non-functional sequence, and explain how it prevents the translation of a full-length protein. And since this is totally new exercise, I would like to help you a bit. So just see the legends here down, okay? So this is the promoter region which is there in yellow. Then the Shine-Delgado sequence has been in this color here. So this is the Shine-Delgado sequence. The start codon is here. Then the coding sequence or gene is here. So gene is in the orange, and then is the stop codon TAA, then is the CCG ATC. So yeah, just have a look at the two sequences. I will give you 5 minutes to tell me what is happening here. So guys, Sudarshan and Akshat, are you able to solve this question? Are you able to undertake this exercise? All right. Yes, Jamil, you're right. But just try to take your time and tell me what would be the implication. Okay, Ria. This exercise is not to test you. This is just to see if you have understood the concepts well. Of course, I will be explaining you um, what is wrong in the first or the second sequence. Okay. So just try to do it by yourself first. just to give you a clue, it is not as complicated as it looks. It is very easy. Just try to focus on this region, okay, here, from the start codon, because you should not look at the common parts which are present. So common parts are not going to make any difference anyway. You might have already recognized it. Let me have a look at the answers. Yes, Akshat, you are partly right. From the live session on YouTube, also someone has answered it correctly. Akhtar, yeah, it is different. Akshat, you're partly right. I would have wanted a little bit of details, but yes, you're right about your answer. I will try to answer this in detail after everyone has taken the time to solve it. What about others? So, Riya, Sudarshana, this is just for your practice, guys, because this is what we actually do in research. If you go wrong here and if you send this particular sequence for to the company for getting the DNA sequence, in the end you will realize that nothing is being done inside the cell, because to start with you send the wrong sequence. So this is very critical. This is the practical part or the application part in the lab. Sudarshana, you are also partly right. I wouldn't say the first sequence is actually faulty, but let's have a look. I think we have taken some time to try to solve the questions. Ria, would you like to attempt? Okay, so Akshat and Sudarshana, thank you for attempting. I am really happy that you tried to solve this, and you are also both partially right. Now, yeah, I will, I will, I will show you. Okay, Ria, no problem. I will show you what is the problem. Okay, so now let's have a look at the answers. So the sequence one is correct. Okay, sequence one is correct. It will lead to methionine, arginine, glycine, alanine, and then the stop codon. And now have a look at the sequence 2. What has happened here is there is a premature stop codon which is present. Okay, immediately after ATG, it is the TAA which is present. So if the TAA is present, it will lead to premature termination of the codon. So premature termination meaning the next amino acid will not be integrated, which means the protein will be truncated. So the flaw in sequence 2 is a single nucleotide substitution. So in place of C, the sequence which is present is T, creating TAA. And we have learned that TAA is a stop codon, which is directly present after the start codon, mean the first methionine which has been produced, and after this there will be immediately a production of a stop codon. So no amino acids will be integrated. This premature stop codon has happened immediately after the start codon, which is why the protein itself has not been formed. It is just a single amino acid. So of course it is a non-functional peptide. Actually, to be very honest, it is not even a peptide. It is a single amino acid. Peptide at least has 3 or 4 amino acids which are present. Okay, so this is not even a peptide, to be honest. So the sequence 2 is faulty. So these are some of the practical aspects. Just a second, Sudarshana sir, in case of first sequence, it is ATG, ATG. What is the effect of the single A base? So Sudarshana, it is already, so even if this A is present, it won't affect it, it will not be affecting it, because the overall reading frame will start from here. So it will not be like ATG. It needs to have several of the factors which are present. So if it is ATG, AA, and if it is even more, two or more of the A's which are present, it will not affect the reading frame. So what matters is what comes also after this. So if ATG has something which is like this, so if this ATG had a GGT and then again we had an ATG, that would lead to a problem. But if it is a starting codon which is ATG and you have another sequence which is present after that, this ATG will be taken as the start codon, not this one. Okay, I hope this is clear. You can all go through these examples again, but it was very interesting question. So I thought you all should be given this question, and you can use a little bit of your intellectual capacity to try to solve this question. So these are some of the things you have to go through it inside a lab when you are doing a cloning experiment, and this will ultimately also decide what you get outside after you do the sequencing, after you do the cloning. And the cloning, as you know, is required as a preliminary experiment in order to obtain any sequencing data later. So yeah, now let's have a look at the real world impact. Like, why does it matter? Why the mutations matter in diseases and evolution? So mutations are responsible for evolution, but they're also responsible for a lot of diseases. So there are many genetic diseases, there are infectious diseases, there are non-infectious diseases. I, being a virologist, would like to walk you through the perspective of virology. So if there are any mutations in the viral genome, for example in case of HIV 1 or 2, which is a very very highly mutable virus, or even in case of SARS coronavirus 2, it can lead to a different viral protein, and this ultimately leads to the escape of the immune system. So the virus is no longer susceptible to the immune system of the host. It leads to changes in transmissibility, which means it can make the virus more infectious or less infectious, and even cause drug resistance. You might have heard about drug resistance in case of HIV 1, also in case of influenza virus. So in the 2009, when the influenza virus emerged, due to the overuse of the drug Tamiflu, many of the viruses were becoming resistant to it. So the doctors were actually telling to prevent the unnecessary use of Tamiflu. So in case of human genetics, so single base changes or indels can lead to different congenital diseases, and this is really devastating. It affects a lot of people globally every year. Now, to show you a practical example, this is a spike protein. So the spike protein of SARS coronavirus 2, which has two domains, so the S1 domain and S2 domain. This is just for your interest, and I thought you would be really curious to know about the real world implication. You don't need to go into the technical details. So the spike protein has two parts, S1 and S2, and this is the original strain which came in 2019. After that, we got a variant A which came in 2021, And after the emergence of this variant A strain, which was termed as Delta strain, there were two mutations which happened, and this mutation uh, continued to evolve. Like, so the virus, when it continued to evolve, this lead to further mutations, and when there were further mutation, it gives to variant B, which was Omicron in 2022. And just ignore whatever is written here. What I would like to point out here is that there is a lot of selection pressure, which means due to the presence of antibodies which are present inside the host and the host receptors, the virus naturally evolves. Okay, and when the virus evolves, there are certain mutations which happen in the spike protein. And what happened in case of Omicron variant is it made the virus even more deadlier, because the Delta mutation and the original virus, which had no mutation, they both only replicated or caused infection inside the lungs. But the Omicron, due to these mutations, was also able to cause gastrointestinal symptoms. So it was also able to damage your gut. So it means that it could damage your lungs plus your gut. So it made systemic infection or infection in multi organs a lot likely in case of Omicron variant than the Delta variant and the Wuhan original strain which was emerging. This is just for your curiosity. So I would like you to go with some important messages from today's lecture. mm, the translation is highly regulated and it requires specific sequences, which are promoters, Kozak sequences, Shine-Dalgarno sequences for the initiation. The protein sequence is strictly decided by the triplet codons between the start and the stop signals. Even change in a single amino acid, single base, that is a missing base or an altered base, can completely change the protein structure or even make it a non-functional protein. And understanding these foundations is very important for molecular cloning, which we are going to learn ahead. For the sequence analysis, because many a times you have to compare two or more sequences. um, I think Biotechshalaa also has several other courses as well as internships where there is a lot of discussion about the sequence analysis. And there are also important examples in the real world diagnostics, because when you want to detect a cancer versus a normal gene, this particular information and knowledge is very important. All right, so this was day 2 of the 7 days molecular cloning basics or molecular master biology class. I would like to remind everyone today again, those who are attending live as well, that today is the live session 2 and is going to be the last live session, which is free for everyone. From tomorrow onwards, all other sessions will be only available for the registered participants. So with that, I would like to open the dais today for any queries or questions. Yes, Sudarshana. The direction of the RNA polymerase reading occurs from 3 prime to 5 prime. Are there questions from the live YouTube session as well? Sir, if we have multiple start codon in one sequence, then which one will be considered as start codon? So the one which is closest to the 5' end will be considered as the start codon. This is also called as the first AUG rule. And also another thing, another rule is that the correct start codon usually has a strong surrounding sequence context. It is also known as a Kozak consensus sequence. Okay, so if it is closest and surrounded by the Kozak sequence, so if an AUG is surrounded by a Kozak sequence compared to the AUG which is not close to the Kozak sequence, then the one which is close to the Kozak sequence will be considered as a start codon. So this is, now you also know why the Shine-Dalgano sequence and Kozak sequences are important. Okay, Akshat. So the actual structure of protein is very complex. So how do we assign names to its different sites or parts, such as the alpha region or beta region? So Akshat, this is just a convention. I think I would talk about it more in detail when we talk about the protein part. But right now it would be a little bit complicated to explain you without any example or figure to explain you. So I think as we go ahead, I can explain to you it in detail. But yeah, there are common things in nomenclature, but there are certain things which are also determined as per just convenience. So there is no strict rule for the determination of regions, but there are of course like different parts to the protein. So alpha helix, beta plated sheets. Does anyone else have any question? Okay, so guys, if no one has any further questions, I would like to once again thank you all for attending today's session. And I would like to end it with a small reminder that there will be two more upcoming assignments which I will upload briefly. The assignments are not mandatory, but I would highly recommend them because you have all enrolled in this course to gain something from it. So in order to gain something from it, you should actually take away some practical aspects, and those will only remain in your brain if you try to solve or answer these questions. So try to solve these assignments, and I'm sure that you will be at least 5 steps ahead than other people who have just learned the concepts inside textbooks. All right, so thank you everyone for attending, and I will end the session for today. Have a good evening, everyone.